Have your lab practical this week? Is that?
Yes.
Okay. All right. It's fine.
No, it really doesn't make sense. I mean, like.
It's like worth less than 10% of your grade. No, all of lab is worth 20%. This one practical is worth less than 10%. No, that's 20%. Each exam is 20%. Yeah. What? Usually phys lab will raise you up. Usually the lab, not necessarily in anatomy. Okay, so things to come, and then we'll go through this. Things to come are your lab practical this week, and then next week, right, is on Thursday is your 4th test, okay? And then on Friday is reading day, which is when I will be in my office for you guys to come look over your tests for the last time. I will have your 4th test with me and your Scantron to give back to you, but I won't have your test graded, right? You'll just have your Scantron. You won't have the other part of it graded, but I'm just going to get them pumped out really quickly so that you can have them to look over and figure out what you missed on your 4th test, okay? So that'll be next Friday, probably 10 to 2, 10 to 3 if I can find somebody to pick up my kid. So like a good chunk of the day, you'll have time. And then your final exam is what day again? Thursday the 8th. So you got, Thursday the 15th. Thursday the 15th, yeah. So next Thursday is the 8th, that's when you have your 4th test. So basically you've got a really fun 2 weeks to look forward to, yeah? It's fine, it'll be over soon. Okay, everybody, yeah? No, it's all Scantron. They have to be graded. You are the last one to take it, and I have to have all the grades in by Tuesday. So if I did, and I have 250 students. So Thursday to Tuesday, they all have to be in. Go ahead.
So we don't have class on the 13th, but Tuesday before?
No. You have any classes finals week except finals, and I have no office hours finals week, so don't even try to find me. I got some crap to do, humans. Go ahead.
If you're on the line of an A, when will we have the 4th test? So we know if we have to take
The fewer questions you ask me, the faster it will get done, generally speaking. Like if you send me 3,000 emails, I am putting Jenny's at the bottom every time you send me an email. I know. You send me emails asking me when your test is done, yours goes to the bottom. How about that? Is that a reasonable explanation? Unless it comes with like money or champagne, then, no, I'm just kidding. Don't send any of that.
Like just don't get A's on the test.
Right. So then we don't have to, we get to relax.
Right after that first one, I'm taking my punch.
Okay, so here we go. We're going to run through this. I want to make sure you understand it big picture, and then we will finish the notes. Okay, so this is the point of the entire story, right? It's blood flow. Hopefully you noticed that when I was trying to be consistent, and I didn't catch them all, but I'm going to re-upload this today, that if there is a positive impact, it was green. If there was a negative impact, it was red. I don't mean negative as bad. I mean resistance tends to decrease blood flow. Pressure tends to increase blood flow. So we know that there is that relationship between pressure flow and resistance. So I want to talk about pressure. We also know that pressure is basically composed of 3 things, right? What your heart is doing, the volume you're dealing with, and what your vessels are doing. So I think Lincoln pointed this out last time. This whole vessels thing impacts both pressure and resistance, right? Because the vessels have sort of that double relationship. I don't try to write questions that confuse you about what the vessels are doing. When I'm dealing with vessels, I will ask a clear pressure question or a clear resistance question, although we know that you could definitely put those 2 together. It's really hard to write a question that can get a decent answer out of it, putting them together. Okay, so let's go here. Cardiac output. Does everybody really understand cardiac output? It's relatively logical. It's a component of 2 things. Stroke volume and heart rate, right? How much per squirt and how many times per squirt, right? Or how many squirts per unit time, right? Okay, so let's look at cardiac output. Let's look at stroke volume. This is the one that people usually have trouble with, so I want to make sure that we get the relationships down before you go study on your own. Stroke volume, a negative impact on stroke volume is something called afterload. Can anybody explain to me what afterload is in your words?
Pressure against the heart.
It's the pressure that you're pushing against, right? Okay, so imagine this. What would happen to my stroke volume if my aorta constricted?
Massive afterload.
Yeah, massive afterload, and it would really drop the stroke volume, right? So it's basically the pressure in the vessels that you are pushing against. If what's happening in the heart is P1, then the afterload is P2, right? And as P2 increases, the stroke volume drops. Does that make sense? Okay, so that's the concept of afterload. This will be really important for hypertension, so we'll come back to that. And then the other two things that can positively impact stroke volume are EDV, which is what gives people trouble, and then the concept of ventricular contractility. So let's break the EDV down. EDV is end diastolic ventricular volume, and it is literally the volume in the ventricles right before they contract, okay, which has two components to it. One of them, the chamber is not a metal box, it's stretchy, so it can be full and it can be fuller, right? And then the other thing is the actual fluid that is in there, right? So you can get it full of fluid, and then you can get it fuller of fluid because you can actually stretch the box. Okay, so both of those things will correlate to EDV. So preload is what is in there, and it's going to be impacted by filling time and atrial pressure. So preload is a direct correlation between EDV. So let's talk about filling time. So the relationship between filling time, so if filling time increases, you have more filling time, right? Then you're going to have more of a preload and therefore more of an EDV, right? And it can increase your stroke volume. So filling time is directly related to heart rate, right? Okay, so if your heart rate is too fast, then you're not going to have enough time for filling, and that's going to drop your stroke volume, right? Does that make sense? Okay, and then the other thing it's related to is atrial pressure. This says that what happens in your atria will impact what goes on in your ventricles, which will impact your stroke volume. Okay, and atrial pressure has two components. We'll come back and add them in just a second. It's basically how full your atria are because of venous return, and then how much they contract. Does that make sense? Okay, so now let's look at Starlings Law. People confuse Starlings Law and ventricular contractility, and I want to make sure that you don't after we finish this. So Starlings Law is that relationship that I talked about on the video of your cardiac muscle sarcomeres. If they are relaxed, they contract relatively well, but if they are stretched, they contract better. And it's actually because they have more access to cross-bridge cycling when they are slightly stretched. So this is not a nervous system phenomenon. This is simply that if you stretch the chamber, then you actually get a better contraction. And it's not elasticity snapback either. It's actually that the sarcomeres work better when they're slightly stretched. It's basically this natural predisposition of the heart to match inflow with output, because it works better when it's stretched. So that's called Starling's Law. It's the length-tension relationship in cardiac muscle cells. And of course, Starling's Law is going to be dramatically impacted by venous return. If the more, you know what venous return is, right? The blood that is coming back to the heart through the veins, which is relatively passive, like not directly related to cardiac muscle contraction. So more venous return will stretch the heart and therefore get better EDV, right? And ways that you can add to venous return are the skeletal muscle pump, right? If your skeletal muscle is contracting, you get better venous return. Heart muscle is better stretched and you get better EDV. But then in addition, the respiratory pump, right? And the respiratory pump is when you are breathing, it changes the pressure between the abdominopelvic and the thoracic cavity, squirts it back up. But these two things are, of course, also directly related to the sort of atrial pressure and preload concept as well. So venous return can actually help with that as well. Okay, go ahead. The respiratory pump is when you breathe in, it increases pressure in the abdominopelvic cavity and comparatively decreases pressure in the thoracic cavity. Yeah. It's that's basically Boyle's law, or is it Boyle's law? Gathol? Can't remember which one it is. Okay, so this is all related to volume and fluids, right? And yes, it says if you stretch more, if you change the volume, you're going to get a better EDV. This is different. Ventricular contractility says hold the volume constant. Don't do anything to it. Leave the same amount of fluid in the same size chamber and then zap it with the sympathetic nervous system and you'll get a stronger contraction. Okay? So this is not volume dependent. This is sympathetic nervous system dependent. Now, how do you get the very, very biggest stroke volume? Well, you actually get as much volume as possible, right? And then zap it with the sympathetic nervous system, right? Does that make sense? Okay, so everybody cool with that? All right, so that's all stroke volume. And then stroke volume is the harder part. Heart rate is not very hard because you've done most of it already because we already did autonomic nervous system, right? So the other way that you control cardiac output is via heart rate. And heart rate is, of course, controlled with the rate of depolarization of primarily the SA node, yeah? And then what controls the rate of depolarization? Well, what controls that is the autonomic nervous system deciding whether to turn on the sympathetic or the parasympathetic nervous system. If the autonomic nervous system, specifically the medulla oblongata, decides to turn on the parasympathetic nervous system, it will decrease the rate of depolarization and therefore decrease heart rate, right? If it decides to turn on the sympathetic nervous system, it will increase the rate of depolarization and therefore increase heart rate. That makes sense? Okay, it will also cause potential release of hormonal epinephrine. The two other ways that you can control the heart rate is hormonal epinephrine, right? It doesn't matter if it was nervous system or hormonal, or thyroid hormone, which will both increase the heart rate. Okay, so the last thing about heart rate is just to put it together with the chemoreceptor and baroreceptor reflexes. So all semester long, we've been sort of implying that the reason you turn on your sympathetic nervous system is because you got a bad job interview or there's a tiger in the room. But now, of course, there are more reasons to do it than that. You use it more frequently than that. So let's talk about this. So gathering sensory information is going on constantly in your blood vessels and in other places. One of them is called the chemoreceptor reflex that occurs in your aortic arch and your carotid sinus. So you have chemoreceptors there, they're sensory receptors, and they are monitoring oxygen, carbon dioxide, and pH. Okay? And so if you assess in the carotid sinus or the aortic arch, any of these stimuli, I want you to tell me which one I should turn on, the sympathetic or the parasympathetic nervous system. If I detect high levels of oxygen, low levels of carbon dioxide, or high pH. Parasympathetic, right. Okay, that makes sense? I have plenty of blood flow. I don't need to turn it up in that particular instance. If I had the opposite stimuli, I'd get the opposite response, okay? And what's the difference between the carotid sinus and the aortic arch? This one's pissier. This one is especially sensitive to pH and carbon dioxide changes because the brain has a crappy buffering system, okay? So it's just, you know, more sensitive basically than this one, okay? That makes sense? Okay, and then the baroreceptor reflexes. The arterial ones make sense to almost everyone. Those are in the same place, but we're talking different sensory receptors here. We have the aortic arch and the carotid sinus, okay? And they are this time monitoring pressure to figure out whether to turn on the sympathetic or the parasympathetic nervous system. The aortic arch or the carotid sinus, if they detected high pressure, then the medulla oblongata would respond by turning on which? The sympathetic or the parasympathetic nervous system? Parasympathetic nervous system, yes? Okay, so this is basically a front-end pressure regulation. If the pressure is too high, then you slow down the pressure in the system. What's the difference between these two? This one is really sensitive to body position changes. If I was laying down and get up really quickly, I start to get lightheaded, and then it will send that low pressure, and it'll say, okay, increase your heart rate, right? This one's not particularly sensitive to body position changes. They can turn on at the same time, or this one can. This one's tweakier. Okay, and then this is the one that can confuse people, so I want to make sure that one makes sense. The right atrial baroreceptor reflex is totally different than these because it's at the back end of systemic circulation. The right atrium, right, is the end of systemic circulation. The superior and inferior vena cava and the right atrium have baroreceptors, but these have a different function. they're really monitoring venous return to figure out what you need to do with your ventricles, okay? So if I have boatloads of pressure in the right atrium, what would make sense? Turn on the sympathetic or the parasympathetic nervous system? Not para. You need to turn, okay, so realize that if I have boatloads of venous return and I turn on the parasympathetic nervous system, my blood's going to start backing up. Because venous return is just going to continue going, right? Venous return is not directly related to what your heart's doing. If you slow down your heart, but you don't slow down your venous return, you're going to back up blood and end up with something akin to congestive heart failure, right? So if I've got a whole bunch of venous return, what do my ventricles need to do? They need to speed up. Yeah, they need to keep up, right? Does that make sense? So that one's the one that's counterintuitive, because you have to realize that you're actually looking at veins but trying to figure out what to do with your heart or your arteries, right? So this one is all lower pressure, right? This is a much lower pressure system than the other one, and it responds counterintuitively. So you have to think through that one. So my job will be to write the clear question. Your job is to read atrial when I write atrial and not arterial, because there is an atrial baroreceptor reflex and an arterial baroreceptor reflex. And usually when I'm trying to write a question like that, I will even put it in bold, but it doesn't mean I can make you read it, okay? I'll do what I can, man. Okay, so everybody good? Okay, so the last little bit is just to remember that we've already learned a little bit about resistance, and so what kinds of things can increase resistance and therefore decrease blood flow? Viscosity of blood, vessel length, right? This one I didn't really talk about very much. If your vessels are elastic, then they tend to be less resistant to flow. If you get hardening of the arteries, which is one of the things that people call arteriosclerosis, then the walls become calcified and it actually adds to resistance, so you get poor flow. And then the other one is vessel diameter, and of course vessel diameter is dramatically impacted by whatever the sympathetic nervous system is doing, even hormonally or otherwise, right? Hormonally or nervous system. Okay, and then last thing is what we're about to talk about right now. The other way of impacting blood flow is by the fluid volume, which is of course primarily controlled by the kidneys, right? Okay, I mean you can sweat some of it out and stuff like that. And the ones that we already have done are these. This one I forgot to make red on yours, and there was another little change that I made, so I'll probably re-upload this. Okay, so which things have a positive impact on blood volume and therefore a positive impact on blood pressure? EPO, right? EPO, erythropoietin, makes you make more red blood cells, so it would be a positive impact on blood volume and blood pressure. ADH, antidiuretic hormone, which comes from posterior pituitary, direct, I was just like, shut up. You all flipped me off mentally, didn't you? So nice of you. Okay, so ADH, antidiuretic hormone, positive impact on blood volume and blood pressure. And then aldosterone, positive impact on blood volume and blood pressure. But we have to learn this little part of the system. That's the last thing that we have to do on that. And I couldn't figure out how to draw it on my computer with, oh, son of a biscuit. So let's do it over here. It's already drawn, but it's better to draw it. Okay, so here's what the kidney does with the renin-angiotensin-aldosterone system. It is in your notes. Show you where it is. We can follow along. You ready? It is right. Where is it? 12? Okay. Oh yeah, right after that. Okay. So the primary way that the kidney controls your blood volume and therefore blood pressure is just deciding how much to keep versus pee. But then there's this indirect way, which is pretty important too. So we'll draw it out so that you guys can sort of get the understanding of how it works. Okay, so here's the deal. I don't know how many of you guys remember the Juxtaglomerular Apparatus from anatomy.
Oh, yeah.
Oh, yeah.
Yeah, so that's generally how I do. You do. Juxtaglomerular Apparatus from anatomy. So So there are these cells, and I'm going to talk about it again in the next set of notes. So there are these cells in the wall of the afferent arteriole leading into the glomerulus, and they're part of what we call the Juxtaglomerular Apparatus, or JGA. Here's what they're doing. The kidney, and specifically it's the JGA in the kidney. If the kidney detects low blood pressure, then that will impact kidney function because the kidneys are pressure driven, okay? So here's what happens when the kidney detects that. The kidney will, those cells in the JGA will release a chemical called renin. And renin is kind of a hormone, but you'll see why it's kind of a hormone in a minute. It's a little difficult to figure out what the hell it is. So it will release renin, and then what renin will do is it will interact with an inactive plasma protein called angiotensinogen. Okay, angiotensinogen is always around. It was made by the liver, and it's an inactive plasma protein. Here's what happens. When renin interacts with angiotensinogen, it becomes angiotensin 1, which nobody really cares about. But angiotensin 1 interacts with another inactive plasma protein. God.
Do you want me to get that while you're...
No. It's okay. Mashed potatoes. I was burning off mashed potatoes right then. Okay, so angiotensin 1, which interacts with another inactive plasma protein called ACE. And ACE stands for angiotensin converting enzyme. Yeah, right? This is what it does. It converts angiotensin from angiotensin 1 to angiotensin 2. Is this a coup? Is this what I'm sensing here? Angiotensin 2. Okay, ACE is always around 2. It comes from the lungs. Now, who cares about angiotensin 2? What angiotensin 2 does is it is the most powerful vasoconstrictor in the body. So it goes to your vessels, causes massive vasoconstriction, which will take care of the blood pressure issue, right? Okay. In addition, it also goes to the adrenal cortex and causes it to release aldosterone. What does aldosterone do to your blood pressure? Raises it. Yeah. Yeah, fluid retention. Sodium and fluid retention. Does that make sense? Okay, so this is why I say that it's kind of a hormone, and where does it come from? So like when I put it right here, angiotensin 2, where does it come from? I don't know where it comes from. This is from the kidney, but this part is from the liver, and that part's from the lungs, so I don't know where it comes from. Kidney, liver, lungs? But it is a hormone, right? Because it gets dumped into the bloodstream, right?
It's strongest vasoconstrictor.
Strongest vasoconstrictor in the body. Okay? Does that make sense? And by the way, it's not that bad. Angio means vessel, tense implies pressure, and gen is the genesis. So this is the beginning, and then angiotensin 1 is converted by angiotensin converting enzyme to angiotensin 2. So the names at least make sense. Good? Okay, this is, have you guys ever heard of ACE inhibitors? ACE inhibitors are blood pressure medication. They use for chronic hypertension to reduce your blood pressure. And if you give an ACE blocker or an ACE inhibitor, it blocks both of these impacts for somebody with chronic hypertension. Good? You're all really happy about it. Okay. All right, so that's the most powerful vasoconstrictor in the body. And then with that, this is the last little part of blood pressure regulation. And of course, it correlates directly to here, this aldosterone. Oh, by the way, if it confuses any of you guys, don't let it. I did tell you before, of course, that you release aldosterone because of potassium concentrations. There's another trigger for releasing aldosterone, and this is it. It's just a different trigger for releasing the same thing, okay? All right. So now what we need to do is change gears for a second. It's really not hard as long as you change gears with me, okay? So what we have just been doing is all of the ways of regulating blood pressure systemically, like as a whole, right? Which would mean, stick with me, if you do anything with the heart, it's going to affect the pressure of the whole system, right? If you do systemic vasoconstriction or vasodilation, it's going to affect the pressure of the whole system. If you impact the fluid volume, it's going to impact the pressure of the whole system, yes? But now what I need to do is switch to how do you impact the pressure and flow to a single capillary bed? Okay, that can't involve the heart because it would jack with everything else, and it can involve fluid volume, right? And it can't involve systemic vasoconstriction or vasodilation. It's just got to be local, arterial vasoconstriction and vasodilation, okay? So this part of the notes is called autoregulation or local regulation, and it breaks all the rules, so you need to set it separate than the other regulation, okay? Because of things like this. If I needed more pressure and flow systemically, what would I do to my vessels? Systemically, constrict. If I needed more pressure and flow to just my homey capillary bed, what would I do? Dilate. So keep that in mind. That's why this goes separate than the rest of it, okay? All right, so autoregulation. Here's what we're going to do. We're not going to try to impact the mean arterial pressure. That's what MAP stands for. It's just the average pressure in the system. We're going to try to hold that constant, but change blood flow to a capillary bed. Okay, we're going to do that primarily with paracrine agents, not nervous system, not hormones. So what happens is I'm going to do local vasoconstriction or vasodilation depending on the tissue's needs. The primary stimulus for this will be the metabolic demands in the tissue. So if I have a capillary bed that is working its little butt off, I'm going to have low oxygen, high carbon dioxide, or low pH, right? And what do you think that that should cause the local arterioles to do? Dilate. Yes. Okay. So this is local regulation. It's independent of systemic factors. It's not trying to mess with anybody else. Could it have slight impacts on the systemic pressure? Yeah, but it's just like secondary. We're not trying to, right? So given a relatively constant mean arterial pressure, the primary stimulus that will cause you to increase or decrease blood flow to a capillary bed is the metabolic demands in the tissue. So if a tissue encounters low oxygen, high carbon dioxide, or low pH, then it will cause paracrine agents to be released, which will cause local dilation of arterioles, leading more blood to the area. Okay? Go ahead.
Where they're released from?
Usually the tissue cells themselves have the capacity to do it. Yeah. And then the other thing, this one's a protective mechanism. This one's a little counterintuitive, but remember that the capillaries are your homies, but they can't protect themselves at all. They have no connective tissue in their wall, and they also have no smooth muscle in their wall. So high pressure will trash them, okay? The arterioles that you have are concerned about that capillary bed only, the arterioles before that. So if they encounter massively high blood pressure, right, they can do what's called myogenic control. And myogenic control tries to protect the capillary beds even at the expense of the system. So what do you think they should do, those arterioles, if they encounter massively high pressure that could damage their homies? No. Constrict. Yes, they protect the capillary beds. Yes, only their capillary beds. Now, could that add to the problem? Yes, but we don't care. Right? These arterioles are only concerned about one capillary bed. So this is like capitalism, right? Like we are only concerned about our little piece, and systemic regulation is socialism. Like everybody needs blood flow. And this one's like, I'm getting mine. That's what's happening here. So myogenic control decreases damage to a particular capillary bed. By the way, if you pit myogenic control against systemic circulation, it will lose. Okay? Eventually, myogenic control will lose. So eventually, if you vasoconstrict, but the rest of the system says that, oh no, the pressure's too high everywhere, we need vasodilation globally, then what will happen is the paracrine agents will release and they'll try to protect the capillary beds, and then they will lose. Because what will happen is you'll release massive amounts of neurotransmitters or massive amounts of hormones. So they'll try at first. Okay, as long as you don't go against the man, you might be able to win. Okay, so now what happens long term if you consistently need more blood flow to an area? Well, you'll start to go through cell division and make more capillaries and make the vessels that you have bigger. So you'll develop collateral circulation over time. You don't do this like the first time you work out, but like the 21st time you work out, you're going to have more blood vessels going to the muscles than the first time, right? Just more blood vessels. Okay, the last thing about this local regulation is just to look at the two extremes. So there are some places in your body that which have very constant needs for blood flow, and some places in your body that have variable need for blood flow. So which one of these two do you think has the most variable need for blood flow, skeletal muscle or the brain? Variable? Skeletal muscle has very variable needs for blood flow. So it's always okay, but then when you start working out, it's massive needs for blood flow. So what happens, how do skeletal muscles bring themselves more blood before you involve the sympathetic nervous system, right? So if you get up and start running, the first thing that happens is as soon as the oxygen level drops in the skeletal muscle, they'll release paracrine agents that cause dilation of their arterioles. But then, of course, you know, the nervous system also starts helping with this. But of course, remember, paracrine regulation is cheaper and faster than the others, so you always try it first. If it was inadequate, then you basically call on your friends, right? You call on the nervous system and the endocrine system, okay? So you get active or exercise hyperemia, which just means way more blood flow during exercise. But the brain is really pissy. Even when you're asleep, it burns almost the same amount of glucose and oxygen as when you're awake. And so it can't have very variable blood flow at all. In addition, it can't have too much blood flow because it's got a rigid cranium. There's no room for extra pressure in there. And then because of its almost zero capacity to do anaerobic cellular respiration, it's intolerant of ischemia, which is a fancy way of saying it gets really pissed off when there's not enough oxygen or blood. So fascinatingly, the main stimulus for autoregulation in the brain is not low oxygen because the brain has such a terrible buffering system, inadequate buffering system, that the brain will cause dilation of its own arterioles leading to the brain as soon as you mess with the pH or the carbon dioxide concentration. And it clicks on way before low oxygen clicks on. So, and then of course the brain is the brain and is the boss, right? So it will bring everybody on board to make sure that it gets enough. But initially it's just as soon as it detects low pH or high carbon dioxide, it'll cause local dilation. Good? Okay. So before we leave pressure, let's just briefly talk about variations in blood pressure, hypo versus hypertension. So obviously pressure is a really important component of flow. There is too low pressure and there is too high pressure. Too low pressure can kill you very quickly, okay? Too high a pressure will kill you slowly and rudely. So let's talk about hypotension versus hypertension. Hypotension is defined as at rest when you have a systolic pressure of less than 100 millimeters of mercury. That's your top number, is less than 100 millimeters of mercury. Does that necessarily mean that you have a problem? No. Mild hypotension can be associated with a relatively long, healthy life, although when you get up really quickly, you'll probably feel if you are hypotensive, right? And you might want to drink a little bit more water and take care of those things. But since it's always difference in pressure between 2 points, if your blood pressure is relatively low, then you don't have to generate as much pressure to go over it. It's easier on the pump and the tubes. But you can get too low. If it's too low that it's not delivering adequate blood flow to capillary beds, it's called circulatory system shock. So shock means something very specific physiologically. It means the pressure is so low you're not delivering enough blood. So basically, since we know pressure, we know there's 3 reasons that you have pressure. There's 3 reasons that you could be in shock. So heart, vessels, volume, right? That's your pressure determinants. So if something happens to your pump, like you had a myocardial infarction, of course you can go into shock because you can't generate enough pressure, right? Or massive vasodilation, like damaging your medulla oblongata that allows you to have vasodilation can cause you to go into shock. Or damaging the sympathetic nervous system can cause you to go into shock. So that's neural damage. But then the other reasons that you might do this are reasons that you might have heard of before, like anaphylactic shock. So what the heck happens with that kid at my kid's school who can't eat peanuts or they'll go into shock? What happens is they have an allergy, and what's going on in that particular situation is you respond to an antigen as if it were a pathogen, and you start releasing vasodilators in response to it, right? So you think you've got some kind of pathogen in your skin, and vasodilation is usually a good response to that, like more white blood cells, more blood flow into the area. But this is not a pathogen. It's just an antigen that you have a response to, and you're not having a minor response to it, you're having a major response. So you take in a peanut and you release vasodilators, they're paracrine agents, but you release so many of them that they get picked up in the bloodstream massively. And so instead of local vasodilation, you do systemic vasodilation and your blood pressure drops dramatically. And you go into anaphylactic shock. Now, how do you treat anaphylactic shock? Epinephrine. And epinephrine is massively effective because we know that it actually causes vasoconstriction, it increases heart rate, but now we also know it increases stroke volume. It's phenomenal at bumping up the pressure, right? That's what it's supposed to be doing, okay? That makes sense? Okay, and then the other reason that you may have heard of this is septic shock or toxic shock, okay? Septic shock, when you have a wound that's dressed, you need to change the dressing regularly or else you're going to cause massive releases of paracrine agents instead of minor release of paracrine agents. Or women always hear about this, and you need to educate your girls with toxic shock and toxic shock syndrome from using tampons. So the vagina's not sterile. There are normal flora in there. If they start to increase in proportion, you are going to get release of immune system paracrine agents, right? And instead of small amounts, you're going to get large amounts. They get picked up by the bloodstream, go systemic, you go into toxic shock. And girls still die of toxic shock. Even around here, people still die of toxic shock. So go ahead.
So this goes back to the anaphylaxis. So if epi is treating like the symptoms, then your body is just left to deal with the allergy itself?
Yeah, so you are only going to have that allergic response as long as you have the antigens present. So if I ate a peanut, antigens are protein-based. Give me a little while, they won't exist in the system any longer, and I'll stop producing these. So if you can get me through the time period in which I had the massive release of these paracrine agents, then I will no longer be releasing them, and they'll get broken down by the liver and the kidneys and all that kind of stuff. So you just have to keep me alive until I'm no longer having the allergic response. Really? Okay, so that's septic shock, toxic shock, anaphylactic shock. They're all basically the same kind of thing and really for the same reasons. Only these two are responses generally to actual living organisms with real antigens, and these are a mistake, a response to something that wasn't a threatening antigen like a peanut, right? Okay, and then loss of blood volume is the other one. If I frickin' bleed out, I'm obviously going to go into shock because I can't generate enough pressure. Does that make sense? Okay, dehydration can also do it though, just so you know. Loss of fluid volume due to sweating, diarrhea, vomiting, diuretics. Biggest killer worldwide of children is loss of fluid, vomiting, diarrhea, diuretics. Okay, so now let's briefly talk about hypertension. So hypertension is defined at rest. This is the last time I looked at the numbers. They may have changed. They change the numbers every once in a while. At rest, if your blood pressure is 140/90, you are hypertensive. Unless, of course, you're sitting there and you're really stressed out. So it's not like you get diagnosed from a single reading, right? So if your blood pressure is consistently 140/90. Let's talk about what happens. So if you go into the doctor and they say, okay, your blood pressure is high, and the person says, why? Well, a lot of times they say it's idiopathic, which means that it's so damn complicated that I don't really want to explain it to you. But generally what happens is it is a dependable progression that starts when you're a teenager. And so it goes like this. Generally speaking, you are going to have a combination of arteriosclerosis and atherosclerosis because of diet and exercise, right? So this is a classic American story right here. So diet and exercise problems lead to arteriosclerosis and atherosclerosis, and you start getting fatty buildup in the wall. It also reduces the elasticity of your vessel, which adds to resistance as well. Add stress into that, and then you get vasoconstriction and water retention associated with stress, right? So what happens is you are increasing T2, right? And so you're dropping your stroke volume, and so what are you going to have to do to maintain the same blood flow? Increase your heart rate. But as you increase your heart rate, you increase your pressure, yes? and then you have to increase your heart rate a little bit more, right? And so this is what happens over time. It's very slight. It's usually asymptomatic for 10 or 20 years. The first symptom is usually not even hypertension, it's high cholesterol, okay? And then later on, you get slight hypertension, but then once you get slight hypertension, then your heart rate starts to go up. So this is the progression of this story that occurs. So why is it negative? Well, it's negative because now instead, I don't know if these numbers are right, but just conceptually, if you, instead of beating your heart, using your heart infrequently but very powerfully, you're going to use it very frequently. And so let's say over a lifetime, you double or triple the use of the same pump. You can see that you will actually be more likely to wear out the system, right? In addition, you're also increasing the pressure on the tubes. Any damage occur when you increase pressure in tubes? You're stretching them, you're potentially scarring them. As you scar them, you're making them lumpier, right? Which makes them more likely to catch platelets. Go ahead.
So once that starts happening, the cholesterol increases, the heart rate increases and stuff, how is it reversed? Can you reverse
The primary thing that you can do is change your diet and exercise patterns, which is the last thing that people want to do. But HDL, remember HDL, our friend? It cleans out your blood vessels. And you only generate HDL by exercising. Damn it! You're like, oh my God, it's the same answer. I'm trying to come up with a question that comes up with a different answer. I haven't been able to figure one out. So yes, it's not a death sentence, but it is a predictive, a very predictable response. So it's asymptomatic for 10 or 20 years, but you are wearing out your heart. The stretching and injuring and scarring of blood vessels leads to more arteriosclerosis and potentially thrombosis, which is catching a platelet and starting the whole intrinsic pathway, right, on accident. You're catching it and starting it on accident. And the increased blood pressure damages your most delicate capillaries. Your kidney capillaries are very delicate, as are the capillaries of your retina and some other places. Do you know that chronic hypertension can lead to blindness? Did you guys know that? And kidney failure. And then, of course, you're increasing your risk of stroke, right, and heart attack, because not only of thrombosis, but also increasing the pressure in these vessels, right? If you're increasing pressure in the vessels, you can also lead to cerebral hemorrhage or cerebral aneurysm, which can lead to stroke. So they call this the silent killer, right? Because it's been going on for 10 or 20 years, long before you're ever diagnosed with anything. So let's talk about risk factors, and then we'll talk about treatment and what you can do about it. So the risk factors are stress, diet and exercise problems, high sodium, high fat diet with lack of exercise. Okay, so if you will exercise, your body will forgive you most things. It will clean out your blood vessels, and you can get away with a third piece of pie as long as you like walk around the block 5 times after you eat it. Smoking is massively important. Smoking, we think that the big thing about smoking is that it zaps the antioxidants from your system, and oxidized LDL sticks to the walls. So I will tell you that from dissecting a whole bunch of cadavers, one of the things that I forgot I had to close down my pictures. One of the things that I figured out about smokers is that although I can't always see the impacts of smoking on their lungs, I mean, I usually can, but not always, I can always see it on their vessels, because this is what smoker vessels look like. That's what they look like. I can take a smoker vessel and I can cut out a chunk of the artery and I can squeeze the fat out of the wall like toothpaste. And they don't even have to be heavy. It just seems to make the stuff glom onto the wall really dramatically. So this is an artery with that blockage in the wall. So the other things that you need to consider is heredity. If you have heart disease or stroke in your family, then you are at higher risk, which means you need to be better to your cardiovascular system. African descent, people of African descent have a predisposition to high levels of cholesterol and arteriosclerosis, which makes heart disease more likely in people of African descent. And then obesity is a big one, because when you have more blood vessels, your blood pressure has to be higher. So you start this whole process despite yourself. In addition, most people don't get obese because they're eating, fruits and vegetables too much, right? So you probably have a couple of factors, right? I mean, it happens, I imagine, but that's not how most people get there. Like, I ate too damn much broccoli and look at me. No, that's not how it gets there. Okay, so what do? The treatment, your doctor, first thing is they're going to tell you to lose weight, restrict your sodium intake to measure the volume a little better. Moderate aerobic exercise, but if you are in really bad shape, make sure you get a consultation from a doctor before you start exercising and blow a gasket. Reduce stress, easier said than done, because I'm stressing you out right now, aren't I? Right? And then there are some treatments, because most people skip straight past this and look to this. The treatments are going to make sense because you either mess with volume, vessels, or heart, right? That's how they're going to regulate pressure. Diuretics are really common for treating hypertension. Beta blockers, the beta adrenergic receptors on the heart and your smooth muscle. If you can block those, you can reduce the sympathetic stimulation on the heart and smooth muscle. And then calcium blockers. I didn't teach you about calcium channels, but calcium blockers block contraction in the smooth muscle of your vessels, so you keep vasoconstriction from occurring. And then ACE inhibitors. ACE inhibitors reduce this whole thing occurring. Those are the most common treatments. What's that? Okay, so that is blood pressure and blood pressure regulation. Okay, so now let's talk about heartbeat coordination and the cardiac cycle. So what we're going to do here is I'm going to go through quickly the stuff that you hopefully learned in anatomy. It's not too hard. And then what I'm going to do is add on to that the cardiac cycle. The cardiac cycle is really this cycle. It's super duper logical. The words sound big, but once you start telling the story, it's outrageously logical. The cardiac cycle is the cycle that goes, when do you contract versus relax each chamber? When do you open versus close each valve? And what does that have to do with the heart sounds? Okay. Before we go any further, do you guys know the heart sounds and what they correlate to? The lubbed up, lubbed up, lubbed up. What? Yeah, valves doing what? Closing, yes. So you can't hear valves open, but you can hear the blood slam against a valve as it closes. And so the lubbed up, lubbed up. The lub is AV valves closing. They're the first ones. It's lower, the sound is slightly lower, and the dup is higher and more staccato, and that's the semilunar valves closing. So keep that in mind as we go through this story. So first thing is just to look at sort of the electrical nature of the heart that most of you guys learned in anatomy, but let me make sure that it makes sense to you. Hold on, I had all this pulled up, but then, okay. Okay, so here is the electrical tissues of the heart, and we already learned about SA node and AV node. But there are other electrical tissues of the heart as well. There is the AV bundle that goes right down here, down the interventricular septum, and then it splits into the left and right bundle branches, and then goes into the Purkinje fibers. So what I want to do with this is just correlate the electrical and the contractile, right? So that you can see how each one occurs. So of course the first thing that occurs is the SA node depolarizes, and as it depolarizes, it's going to send all of those ions to its buddies in the atrium. So the atria are going to contract in what order? Top down, right? They're going to contract top down. In the order that they got the stimulus is how they're going to contract. Okay, but by the time you get to the bottom of the atrium, the AV node was on its way to depolarization. It spontaneously depolarizes too, just not as quickly as the SA node. So it was already going to do it, and it will do it, just so you know, if the SA node doesn't tell it what to do. It was on its way, and then the SA node floods it with ions and says, do it now. It's like you're on your way out the door to take the garbage out, and then your mom says, will you take the garbage out, and you lose all the credit, right? I do it all the time to my teenagers. Well, actually both times that they took the garbage out spontaneously. Both times. My son will do it. Teenage girl, goodness gracious. Okay, so this is the AV node, but the AV node does not talk to contractile cells, and this is really important for you guys to understand. If it started talking to contractile cells, then I would contract here first, right? And then up here and then up here. That's not how I want the ventricles to contract. So what the AV node does instead is it talks to the AV bundle, which is still pacemaker cells, right? And then the pacemaker cell still doesn't talk to contractile cells. It talks to the left and right bundle branches, which are still pacemaker cells. And the left and right bundle branches do not talk to the contractile cells either. It's only, they talk to the Purkinje fibers, and then the Purkinje fibers talk to contractile cells, okay? That means that there's a forced lag time between atrial contraction and ventricular contraction. It also means that the ventricles should contract in what order? Bottom and sides up, and that's what they do. So what's important about that forced lag time between atrial contraction and ventricular contraction? Filling, yes, exactly. That's when ventricular filling occurs. At rest, there's usually about 0.2 of a second between atrial contraction and ventricular contraction, right? So you got time for that thing to fill before it contracts. That makes sense to you guys? Okay, so now, what does the whole autonomic nervous system do to this? Really, it doesn't do anything except change the rate, right? SA node depolarization rate, AV node depolarization rate. And when all things are good, the AV node always follows what the SA node does. If it doesn't, then you're going to have fibrillation or abnormality of conduction or contraction. Okay, so that's how it's supposed to work. It doesn't always work that way. An arrhythmia is anything other than the heart doing atrial depolarization, contraction, ventricular depolarization, contraction. This, right? An arrhythmia is just anything other than that. So there are a couple of types of arrhythmias. There are a bunch of types of arrhythmias, actually. I'll teach you a couple of them, and then I have an adorable video to show you some more of them. So the fibrillation is one type of arrhythmia. Here's what a fibrillation is. If you have an atrial fibrillation, then the atrium, that's not it, yeah. If you have an atrial fibrillation, then the atrium is doing this. Okay, this is what it's supposed to do. I'm not very coordinated, so this is the best I can do. Okay, so this is what it's supposed to be doing. Atrial contraction, ventricular contraction. Atrial contraction, ventricular contraction, right? When you have atrial fibrillation, the ventricles are totally cool, they're doing their job, but the atria are spasming out. That's what my particular heart problem is. Atrial fibrillation, it goes spaz out. And then when you have ventricular fibrillation, it's when the atria are doing what they're supposed to do and the ventricles are spazzing out. Which one's going to kill you real fast? Ventricular fibrillation or ventricular tachycardia. Atrial fibrillation can kill you. It's not going to kill you dependably. It will drop your blood flow by about 20% because the atria have to contract to push the last 20% into the ventricles, which I can attest will make you pass out on a bad day. Most of the time it just makes you kind of lightheaded, but the big risk there is that the blood's just sitting in the atrium instead of flowing, and it increases the likelihood of clotting dramatically. So if you have a decent bout of atrial fibrillation, you're at risk of heart attacks and strokes for about 2 weeks afterward. So the whole time I was pregnant, estrogen increases your likelihood of clotting. So the whole time I was pregnant, I had to shoot myself up with anticoagulants twice a day because of the combination of that and estrogen.
What if you had such a bad time when you were pregnant?
Oh yeah, it was. I ain't doing it again, man. I like check that one off the list. Ain't happening. Okay, an ectopic pacemaker is when someone other than the SA node tries to be in charge. This is a coup, right? And then what happens is, oh, I've got the right ventricle that's just trying to do its own damn thing, and everything else is okay. Or the ventricles are trying to contract before the atria. That's what an ectopic pacemaker is. Ectopic just means out of place. Okay, so these adorable cardiologists made a heart rhythm dance for you guys, and I know that you're going to enjoy it. Oh, well, shut up then. Just sit there and look amused. They're so cute, aren't they? It's like somebody's dad. I'm sure their daughters are mortified. Atria on the top, ventricles on the bottom.
Wow! Bonjour, ma chère. Tu as bon invited. Comme cela était l'île, la mer, la plage.
This is what I've got. It's about as well as I dance, too. So cute. He's adorable. This is ventricular tachycardia. This will kill you. See? Yep, goes back in. I like this one. I can already, I know it's okay. I know. Gets pretty boring in those labs at night. Okay, so the last thing is we are going to put together the cardiac cycle. It's really easy. It doesn't sound easy the first time you go through, but it's super logical. So I'm going to show you the figure in your textbook first, and then we're going to look at the animation. Okay, so this figure looks outrageously complicated. It's one of those figures that you're better off looking at home, but it's good. Okay, so here's what I want you to look at. There are going to be a couple of different places. What we're trying to keep track of here is when the chambers contract versus relax, when the valves open and close, and where the blood is going. This whole thing is going to be a cycle, and it's going to be called the cardiac cycle. So let me introduce you to the terminology of it, and then we'll start to add in the details. There's some logic that makes it really easy. Once you get started in one place, you don't really have to memorize it. It's pretty damn easy. Okay, first off, I'm going to start you with the first stage. The first stage is called ventricular filling. Okay, ventricular filling. Now, ventricular filling, I don't probably have to tell you the position of the AV valves during ventricular filling, do I? AV valves open or close during ventricular filling? Open. And just so you know, in a normal heart, you don't have to memorize what the other valve is doing. If one of them's open, the other one's closed. Okay? So if the AV valves are open, the semilunar valves are closed. Does that make sense? So I've got ventricular filling. Okay. Now, to move forward, ventricular filling, at the end of ventricular filling is when atrial contraction is going to occur. That means the AV valves open before the atria actually contract. They open because of venous return, not because of atrial contraction. Okay, but now conceptually, ventricular filling is going to end when what valves do what? Not yet. AV valves have to close. Yeah, so what's going to happen is the ventricular filling occurred, and then the ventricles are going to start to contract, okay? And then AV valves are going to close. That will be the end of ventricular filling. Just conceptually, it has to be, right? You can't fill the ventricles anymore. I'm going to have this brief period. Look at the length of the period. This is the relative lengths of the periods. This brief period that I just closed the AV valves and the pressure is still increasing and I haven't opened the semilunar valves yet. This is an important period because it keeps the valves from ever being open at the same time. This period right here is called isovolumetric, because no fluid is moving anywhere, ventricular contraction. You with me? Okay, isovolumetric ventricular contraction. Now, isovolumetric ventricular contraction is going to end when what valves do what? Semilunar valves open. Yes? That starts the period of ventricular ejection. Again, you don't ever have to memorize the condition of the other valves. If the semilunar valves are open, the AV valves are closed. Yes? So this period of ventricular ejection is distinctly longer than that isovolumetric period. Do you see that? Okay, so now think about this. I'm doing ventricular ejection. Ventricular ejection is going to end when what valves do what? Semilunar valves close, and they close because of the drop in pressure in the ventricles will make the blood try to go backwards, right? And it gets caught in the cusp of the semilunar valves. Then I have this brief period right here when the ventricles are relaxing, all the valves are closed, no blood is going anywhere. This is called isovolumetric ventricular relaxation. Good? Okay, and that's going to end when what valves do what? AV valves open, exactly. That's due to venous return. So, by the way, this one right here is just the start of the same story over again, okay? Okay, so ventricular filling At the end of ventricular filling, I have atrial contraction, and then you start the whole story again. Okay, so notice that if you look at this, you can at any point tell what the position of the valves are, but do you see that if you start it logically, you don't have to memorize that crap, right? You just like start it and understand what the words mean, and then just follow it until you get back there again, okay? I'm going to show you an animation too. We can also put together the heart sounds with when these things occur, because the lub is at the end of, it's when the AV valves close right here. It's right at the end of ventricular filling, right there, okay? And then the DUP is where? Right here. The semilunar valves close, right? Okay, so I'm going to show you an animation that will put it all together in one story. He uses isovolumic instead of isovolumetric, which is what your textbook uses, but it doesn't really matter. It's fine. Okay. And this animation is posted for you on the links page of melanieb.com too. Cardiac cycle. Okay. Okay. So let me just show you where he's starting. Can you tell where he is right now? Ventricular filling. The atria are starting to contract, so this is the end of ventricular filling, right? Okay.
Ventricular contraction causes the atrioventricular valves to close, which signals the beginning of ventricular systole.
This is isovolumetric ventricular contraction. Yes? And it's going to end when what valves do what? Semilunar valves open. Yes.
The semilunar valves were closed during the previous diastole and remain closed during this period. Continued ventricular contraction increases pressure in the ventricles above the pressure in the aorta and pulmonary trunk, causing the semilunar valves to open.
That's ventricular ejection. Good.
When the ventricles relax and their pressures drop, blood flowing back toward the relaxed ventricles causes the semilunar valves to close, which is the beginning of ventricular diastole.
Isovolumetric ventricular relaxation.
Note that the atrioventricular valves remain closed. When the pressure in the ventricles becomes lower than the pressure in the atrioventricular valves open and blood flows into the relaxed ventricles. This accounts for most of the ventricular filling. The atria then contract and complete the ventricular filling.
So ventricular filling, isovolumetric ventricular contraction, ventricular ejection, isovolumetric ventricular relaxation, ventricular filling, right? And then atrial contraction, and you start the whole damn thing over again, right? So this is the cardiac cycle. Do you see that? You know, if you can get started, you can do it. And all you have to do is be able to remember the names. Okay, does that make sense? Okay, so last little tidbit before we leave is this is all written in your notes as well. You guys did EKGs or ECGs in lab. I just want to take a minute to put together the concept of ECGs and what we're talking about here with the cardiac cycle. Okay, so the mechanical activity of the heart is determined by the electrical activity of the heart, right? So the depolarization will cause contraction, and then the pressure changes will cause valves opening and closing, yes? Okay, so let's see if you guys can do this. The electrical event is atrial depolarization represented by the P wave. My question to you is, what mechanical event occurs because of that? Atrial contraction. And then the next question is, does that mechanical event, atrial contraction, cause the opening or closing of any valves? No. No. It is not atrial contraction that causes the opening of the AV valves, it's venous return. So that's why I can live with atrial fibrillation, right? Does that make sense? If I was waiting for my atria to contract fully to open my AV valves, then I would have died by now, right? But they actually open because of venous return, right? That's why that last squish of blood is only 20% of the blood moving in, not 100% of the blood moving in. Does that make sense? Okay, so the atrial contraction does not cause the opening or closing of any valves. The next electrical event to talk about is ventricular depolarization, represented by the QRS complex. Yes? And the mechanical event that occurs because of ventricular depolarization is what? Ventricular contraction. And now, same question. Does that mechanical event, or the pressure changes because of that mechanical event, cause the opening or closing of any valves? Do them in order. There's two. It does two things. Close AV and then open semilunar. And it's not the contraction that causes it, it's a pressure difference because of the contraction, right?
Yeah,
Okay.
I was just gonna ask.
Yeah, it's really a pressure change, but the pressure change is generated by contraction of the ventricles, right? Because it's a closed chamber and you're squishing on a closed chamber, so it closes the AV valves and then opens the semilunar valves. Does that make sense? Okay, so on Thursday we are going to do urinary system, and then probably on Friday I will post a little pre-lecture for the respiratory system so that you're not as overwhelmed. It will be the same amount of material, but if you have more time to digest it, you will do better on it.
So it's not joint.
I like doing urinary system next. I hate respiratory system, honestly, so I'll just do it last.
Wait, so it's best class.
Urinary system, huh?
Oh, Thursday, we were.
Thurs-
Tuesday.
Thursday, urinary, Tuesday, respiratory. Yes. If we have time. Depends on whether we have time. Hold on, let me save this real quick.